Good morning, everyone. If you're in the US, good afternoon for those of us in Europe, wherever you are around the world. It's a great pleasure to welcome you to the PGC MDD's takeover for the Worldwide Lab meeting this afternoon. And what we've done is we've taken the opportunity to introduce you to the PGC MDD group, and here we are at our last meeting at the World Congress of Psychiatric Genetics in Anaheim at the end of last year, and I think we are all rather disappointed that there won't be a similar photo in Florence this year, but we will still be meeting and our science will still be moving ahead as always. So what we're going to do in this session is introduce you to some of the early career researchers that form the PGC MDD group. And that's really where the powerhouse of research is, not just in our group, but in every group. And so we have invited 6 of our early career researchers to give you an introduction to depression, going right from the beginning, the disorder and the epidemiology, and walking us carefully through to the genome-wide association studies, genetic correlations, and gene-environment interactions. So I'm hoping that this session will be a really good introduction to both the people giving the talks and the excitement that's currently going on in depression as we find more and more about the genetic predisposition. So I'm going to, I've stopped sharing and I'm going to hand over to our first speaker, who's Kimberley Kendall. So Kim is a clinical research fellow at Cardiff University. And she's been studying neurodevelopmental CNVs across the phenotypic spectrum, not just in depression, for her PhD, and she's shortly heading back to clinical training. And for the MDD group, she's our liaison with Jonathan Seabat and the CNV group, and we're really looking forward to her work with that. So Kim is going to kick off this session with a talk on an introduction and epidemiology of major depressive disorder. Over to you, Kim.
Thank you, Cathryn. Good afternoon, good morning to everyone. I'm going to talk through an introduction to the major depressive disorder phenotype and its epidemiology. Depression is a common mood disorder with the potential to have substantial impacts on an individual's morbidity and mortality. Globally, depressive disorders are ranked as the single largest contributor to non-fatal health loss. The 12-month prevalence of depression is around about 6%, but this does actually vary quite considerably according to country. And the lifetime prevalence is a lot higher at 15 to 18%. The disorder is twice as common in females as males, and this phenomenon is termed the gender gap. Relatively speaking, major depressive disorder is a disorder of younger people. So the most probable onset period is from mid-adolescence to mid-40s, but almost 40% of people with this disorder will have their first episode before they're 20 years of age, and the average age of onset is mid-20s. Major depressive disorder is characterized by the core features of depressed mood, which need not manifest as sadness. It could also manifest as feeling irritable or empty. And a symptom of anhedonia, which is an inability to experience pleasure. But in reality, most patients will have additional symptoms from the emotional symptom groups, such as feelings of worthlessness, neurovegetative symptom groups, such as loss of energy or alterations in sleep and appetite, and the neurocognitive group, which is things like an inability to concentrate. These symptoms can vary according to the severity of the disorder. So for example, you would tend to be more likely to see something like psychomotor retardation in someone who's more severely unwell. And the phenotype itself is very heterogeneous. The disorder can vary in terms of clinical presentation, onset, course, and severity. And a study of major depression symptom profiles found more than 1,000 different profiles in a sample of individuals with the disorder. I've shown you a table here with the 10 most common symptom profiles. And what I think is quite stark is that the most common profile was only endorsed by 1.8% of the participants. So this heterogeneity can make it quite difficult to manage clinically and to study, although I would argue that this also makes it more interesting. There are several established risk factors for major depressive disorder. I'm a psychiatrist, so I tend to approach it from a biological, psychological, and social standpoint. Biological risk factors include, obviously, genetics, and more on that later. Some of the psychological risk factors include things like adverse life events, such as bereavement, and the social risk factors include things like low income and socioeconomic status. There's substantial comorbidity between major depressive disorder and other psychiatric and physical health phenotypes. In psychiatry, probably the best known one would be with anxiety. But we also know that there are other psychiatric disorders which have a high risk for depression, for example, schizophrenia and autism spectrum disorder. In terms of physical health, individuals with major depressive disorder have an increased risk of various physical health problems, including asthma, gastric ulcers, hypertension. And we also know that it works the other way around. So individuals with chronic physical health problems have a marked increase in their likelihood of developing depression. The nature and the direction of these associations can be difficult to establish and untangle. And there's also the question of whether the depression you see on a background of, for example, physical health problems is phenotypically and biologically the same as the depression you would see in what we consider to be the syndrome of major depressive disorder. And I think that's quite an interesting question and something that can be unpicked in the future. So in summary, major depressive disorder is common. It's a disorder. Also, obviously, it affects older people as well, but onset tends to be when people are younger. It's characterized by low mood and anhedonia, but there are lots of other symptoms and it's really very heterogeneous. There are quite a range of established risk factors and also high rates of comorbidity, but untangling associations can be tricky. Thank you very much for listening, and I'll now hand you over to Luykx.
Hi, so it's a pleasure to introduce Luykx, our next speaker. Luykx is Assistant Professor at the Karolinska Institute, working on the genetic and environmental contributions to depression, and has just been awarded with Pat Sullivan an NIH grant to look at a trans-Nordic study of extreme major depression. She takes responsibility for secondary analysis proposals in the PGC MDD group and is going to speak to the title Genetic Epidemiology of MDD. All yours, Luykx.
Yeah, can you see my presenter view or?
Yes. Yes, we can.
So it's all good. It's all good, Luykx.
Okay, all right. Thanks, Andrew. I will talk about genetic epi. So it is clear that MDD is a complex disorder, so neither gene or environment plays their role alone. Early evidence suggested that MDD runs in the family with an increased risk of nearly threefold in the first-degree relatives. But how much is it attributable to gene or environment respectively? The answer to this question will guide our current gene mapping effort. Heritability is a key concept in genetics. It is a proportion of phenotypic variance that's attributable to additive genetics. It is typically estimated from twin studies based on the expected genetic and environmental sharing within the twin pairs. There is a long history of twin studies. Here, I will go through a few key papers. First is the meta-analysis from 2 decades ago, and yet this estimate of 37% is still the most cited MDD heritability to date. Next is the single largest twin study of 15,000 pairs. And the third one is the largest meta-analysis of all twin studies in the past 50 years. The last one is a more recent study. It had a higher, slightly higher estimate, potentially due to the higher severity in the treated cases. Overall, the heritability from twin studies are fairly similar. But twin studies do rely on a few key assumptions, especially the same shared environment assumption between the two twin types. This was relevant for the missing heritability debate because some suspected that these twin heritability might have been overestimated. So what are other lines of evidence? There is a growing literature of heritability estimation based on the population-based registers or EHR data. These studies reconstructed extended pedigrees based on either recorded or inferred familial relationships. To estimate, these estimates look quite similar to the twin studies, so there seems to be limited evidence for overestimation. Now, what about the environment? First, about the shared environment. So here the twin studies seem to be a bit inconsistent. However, unlike the twin study, the third one looked at the siblings reared apart and together, so it doesn't rely on the common shared environment assumption. Therefore, this study might provide a more accurate estimate of about 5% or less due to the shared environment. So this leaves up to 70% of variance to unique environment, making it the largest variance component for MDD. Next question, is there a sex difference in the MDD heritability? The motivation there is to see if the higher female prevalence can be explained by a higher genetic component. As you can see in the figure, the heritability does seem to be higher in females than in males. But early meta-analysis failed to detect a significant difference, whereas the other twin studies showed a genetic correlation between the sexes much lower than 1, although with quite large confidence interval. The more recent study based on the siblings also was able to provide a more precise estimate of 0.9. So despite a higher MDD heritability in females, the majority of genetic risks are still shared across sexes. And the heritability difference between males and females doesn't seem to fully explain the female preponderance in the phenotype. Now, given the relatively low heritability of MDD, it will be extremely useful if we can identify clinical subtypes that are more heritable. One successful example is the Converge consortium, where they ascertained recurrent MDD cases in women to enhance GWAS power. Early evidence only able to establish recurrent MDD as a more heritable type. However, the recent studies based on large-scale population data was able to extend the evidence to early age of onset, comorbid anxiety disorder, those with greater clinical severity, and postpartum depression. Now, to summarize, MDD heritability is about 30 to 40%, but it's important to note that heritability is population and time specific. There are sex differences and subtypes with higher heritability. And finally, the combination of high lifetime prevalence and low heritability makes it really challenging for gene mapping. And the next 2 sessions will be about that. Thanks.
Thank you very much, Louis. You probably have gathered by now that what we've decided to do is to roll through all the talks at once and then have a longer Q&A session at the end. But if you want to write your questions in the Q&A box at the bottom of your screen while you remember them, that's a good idea, and then we'll come to them all at the end. So for our third talk, it's my pleasure to introduce Eva Schulte, who's a clinical research fellow in the Department of Psychiatry at the University of Munich. And in her PhD, she worked on rare variants in common complex neuropsychiatric disorders, which ties in with her talk title here. And she's now moved on, switching her focus to lipidomics in psychiatric traits for her postdoc. Eva, I'll hand over to you.
Thank you, Cathryn, and it's really great to have you all in Munich today. Welcome. Once people had identified that there was a heritable component to MDD, they actually set out to try to identify that underlying genetic component. For the first 40 years, since about 1978, people did so by performing candidate gene studies. And most of the more than 200 candidate genes that were assessed belong to a number of neurobiologically implicated pathways, which are shown here on the left, and that amounted until 2014 to more than 1,500 individual studies. Since most of these genes and variants were assessed in more than one study, people were also able to meta-analyze and show that 7 common variants were significantly associated with the MDD phenotype. However, overall, data were very inconsistent and the power of these studies was highly limited. So now that we have transgressed these 40 years of candidate gene analyses and have arrived in the whole genome era, what do we think of candidate genes? GWAS currently lend no support to the classical candidate genes or the pathways that they're implicated in. And a very recent systematic analysis of the most commonly studied polymorphisms in 18 candidate genes in both the UK Biobank and the PGC's MDD group data, looking at quite a number of different samples, found that there was no evidence for association with several different definitions of depression. So not just MDD itself, but also self-reported depression, etc. So overall now there's no genetic evidence that variants in candidate genes contribute to the context of depression. And most of the authors of the current studies actually suggest that we stop looking for it. Most of the variants assessed in the candidate genes were actually common variants because really that was the only thing people had access to at the time. However, there is one big conundrum in that common variants only harbor small effect sizes, and therefore you need very large numbers in order to statistically significantly associate them with a phenotype. Nowadays, we do know common variants that are significantly associated with MDD. However, they only explain a portion of the heritability, and there is still missing heritability, which may also lie in a collection of rare variants of larger effect sizes. Although these may not play a role in all individuals with MDD, they might be there in some cases, and those cases are interesting because rare variants harbor larger effect sizes, and therefore these variants are much more amenable to studying the underlying biology of the disease. Nowadays, rare single nucleotide variants are studied using whole exome sequencing in most cases, although at least I'm not aware of any large-scale efforts just comparing cases and controls in MDD that are currently going on, just because you need very, very large numbers. And people have tried to circumvent this by assessing subphenotypes. So for example, looking at sex differences, suicide completion, treatment resistance, family studies, early onset MDD, and also certain MRI changes. While of course it is very difficult to summarize all these studies in one single sentence, what I did find striking is that most of them reported an enrichment of singular rare or low frequency variants in specific cases, but none of these really reached statistical significance, and also basically all of them lacked replication. So at this point, there's still quite a bit of work to be done to finally or conclusively implicate any single nucleotide rare variants in the genetic architecture of MDD. Next to the single nucleotide variants, copy number variants are another form of genetic variation that has been shown to play an important role in neuropsychiatric diseases. However, with regard to MDD, there are currently very few high-powered studies that have been performed, although, as Cathryn already mentioned, there is a large ongoing effort to rectify that from the PGC and also the MDD group within PGC. One study that was performed sort of on a large scale showed that there was an increase in genome-wide burden of rare short deletions in MDD, and these were largely localized to intergenic regions and enhancer regions. And Kim, who actually presented the introduction, was also able to show that 53 copy number variants of known association with neurodevelopmental phenotypes are also associated with self-reported depression. Yet at the same time, it is important to note that currently there is no evidence for any large-scale multigenic CNVs in the genetic architecture of MDD, such as those that have been implicated in schizophrenia or autism spectrum disorders. To summarize, genetic variation in candidate genes does not seem to contribute to the genetic architecture of MDD. For rare single nucleotide variants and CNVs, most studies are still too small to actually draw any final conclusions on how much or if at all rare variation contributes to heritability. Still, it is important to look for these rare variants because they are likely to harbor larger effect sizes and are therefore better able to inform the underlying biology. And at least to me, that is sort of one of the ultimate aims in studying MDD genetics in the first place. So thank you, and I'll hand over to Till, who will talk about the common variants.
Thanks very much, Eva. Yes, Till Andlauer is our next speaker, and he works in the Department of Neurology at the Technical University of Munich, working on a range of genetic studies in multiple sclerosis and psychiatric traits. So Till is going to speak to the title Genome-wide Association Studies. So over to you, Till.
I was muted. That didn't really help with my talk, but I think now it's better. Yeah, so I'm going to talk about GBAS, and the first GBAS conducted between 2011 and 2015, they identified only up to 2 different significantly associated regions. And this changed from 2016 on, when the sample sizes increased, and we're now in the latest published study in 2019 at 102 independently associated regions. And these are mainly due to the efforts of the PGC in recruiting more samples, but also due to 23andMe and UK Biobank, who contributed large cohorts. I'm gonna tell you a bit about the biology of what was found in these GBAS using the study from 2018 by the PGC that analyzed 135,000 cases from the PGC and other studies. Notably, about half of the cases were from 23andMe and thus included cases of self-reported depression. In this study, 44 genome-wide significant loci were identified. And it's always difficult in a GBAS to go from the associated SNPs to implicated genes. But when using a positional overlap at the associated loci, one could observe that many of the putatively associated genes play roles in neurite outgrowth, in synaptic function and plasticity, but also in immunity and inflammation. In this study, also magma-based gene level and gene set analysis were included. And in these gene level analyses, many genes were implicated that code for pre- and postsynaptic proteins, especially receptor subunits like voltage-gated calcium channels, dopamine receptors, and several different glutamate receptor subunits. And also the pathways that were found that point at functions in neuronal differentiation and protection, at the immune response, and mainly at synaptic function. And the synapse, especially the postsynapse, that really appears to be a key role for the development of depression, but also for other psychiatric disorders like, for example, schizophrenia. And here it's really synaptic plasticity with all of its players like voltage-gated calcium channels, then RNA binding proteins like FMRP that regulate the local translation and regulate how glutamate receptors are incorporated and removed from the synaptic membrane. All of this appears to be very important for the development of psychiatric disorders as confirmed in these GBAS. And last year, already the next GBAS was published that included mainly more cases from 23andMe and UK Biobank, and that increased the list of significantly associated loci to 102. Biologically, it mainly confirmed and extended what was already known from the previous GBAS. And interestingly, using these GBAS, we can also calculate the polygenic risk scores for MDD. And when using that, to predict the case control status of depression in independent cohorts. I have to say the prediction performance is rather poor. So the pseudo R-square, the maximum achieved was about 3%. That's not too great yet. Just a month ago, another preprint was uploaded to medRxiv with an even larger GWAS centered around the Million Veterans program. They now identified 178 associated SNPs. And this study included a trans ancestry analysis of European and African ancestry samples. So to sum up, we now identified dozens of different associated loci, but each of them have very, very small individual effects. So the maximum odds ratio observed was 1.05. That's really tiny. The genes that were identified, they indicate that pathophysiological mechanisms involving neuron development, synaptic plasticity, and inflammation are important for depression. And many of these implicated genes, or at least some of them, they are also important for other psychiatric disorders, for example, schizophrenia. And you may wonder that in studies, for example, on schizophrenia using comparable sample sizes, much more genome-wide significant risk loci were identified than for depression. And likely this difference is due to the higher prevalence and lower heritability of depression, as already pointed out, and likely also because of the more heterogeneous patient samples. And this heterogeneity really matters because the phenotype definition is important. The PGC mainly use diagnostic interview, but for example, 23andMe self-reports, and depending on how the phenotype was determined, you get different GWAS results. So this is something important to keep in mind. And with this, I'm at the end. Thanks.
Thank you very much, Till, for that introduction to our genome-wide association studies. And then it's been amazing to see how the number of detected loci has gone up over just the last few years. Our next speaker is Jurjen Luykx, who is at the University Medical Center Utrecht, where he's a psychiatrist, and he performs a wide range of clinical studies across all the psychiatric disorders. And he's going to tell us about genetic correlations with MDD. Jurjen, over to you.
Thank you, Cathryn. So I'll briefly discuss the genetic correlations that have been found for MDD over the past decade or so, maybe less. First, to start, what did I do to get to these genetic correlations? First, like a brief introduction to what exactly genetic correlation is. It's a statistical measure of correlation between genetic underpinnings of one trait and genetic underpinnings of the other, and it can range from 1 being perfect positive genetic correlation to -1, which is perfect negative. And so I extracted the genetic correlation results from recent genome-wide association studies where I focused on the largest GWAS for MDD, but also some other studies that specifically delved into other kinds of phenotypes to see whether there's genetic correlations. So first is maybe a graph you're familiar with. It's derived from the recent Cell Cross Disorder paper that came out last year, where basically what you can appreciate from the graph is that MDD is almost right in the middle, grouping with bipolar disorder and schizophrenia on the one hand, and also with autism spectrum disorders and ADHD. And it's associated with 48%, so almost half of the 109 pleiotropic loci that have been found across these 8 main psychiatric disorders. So I think first to show a graph from the Brainstorm paper where psychiatric and neurological disorders were compared and genetic correlation analyses were run. Basically, there's very few genetic correlations between neurological and psychiatric traits, except for MDD and migraine and ADHD and migraine. For the rest, there's very little overlap across neurological traits and also between MDD on one hand and other neurological traits. So I think this is an important graph to show the main genetic correlations that have been found in the 2 largest GWAS studies that have also been discussed by Till. So on the left, you see the Ray et al. Nature Genetics 2018 paper, where on the top you see that depressive symptoms, not surprisingly, list very high has a genetic correlation of almost 1, 0.9. And on the right panel, you see a graph from the Howard et al. Nature Neuroscience 2019 GWAS, where basically depressive symptoms were also ranking highest. Not surprisingly, I think for a psychiatrist, neuroticism also ranks really high. And when you look at genetic negative correlations on the right, you see that subjective well-being, college completion are negatively associated with MDD. Another study that I found of interest is a study relating different kinds of mood disorders, such as bipolar 2 disorder and bipolar disorder type 1, and different kinds of major depressive disorders, so recurrent, single, and subthreshold depression. And what you can appreciate from this graph, where the genetic correlations are also depicted in these numbers, is that recurrent major depressive disorder has a way higher genetic correlation, more than double with bipolar disorder type 2 than with bipolar disorder type 1. And I think as a clinician, I think you can basically understand, because it's also apparent in the clinic, where sometimes bipolar 2 disorder has less clear manic episodes, hypomania, whereas bipolar 1 disorder has clear manic episodes, so being kind of a distinct phenotype. So I think this is kind of confirming what we also see in clinical practice a lot. And obviously recurrent major depressive disorder, single episode major depressive disorder, and subthreshold disorder, depression, have very high genetic correlations if you compare them to other mood disorders. And then atypical symptoms. So these are symptoms that are not commonly seen in most depressive episodes. Maybe they're observed in about 10 to 20% of major depressive episodes. So these are increased appetite and increased weight. So if you look at the graph on the right, this is a genetic correlation graph with BMI. And what is apparent is that this subtype of MDD with atypical symptoms, so increased appetite and weight, is strongly genetically correlated with BMI, whereas other subtypes of MDD, decreased appetite, for example, are not associated with BMI. So in conclusion, genetic correlations for MDD are strongest for mood and personality-related traits, being social well-being, depressive symptoms, and neuroticism. And with that, I would like to conclude, and thank you so much for your attention.
Thank you very much, Jurjen. We'll move on now to our next speaker, Karmel Choi. Karmel Choi is a research fellow at the Harvard T.H. Chan School of Public Health and Mass Gen Hospital, where her research focuses on the interplay of genetic and environmental factors that influence trauma and resilience across the life course. She is the outreach representative for the PGC MDD group, and you should look out for the new PGC website coming soon. So Karmel's going to speak to the title Gene by Environment Interactions. Over to you, Karmel.
Thanks, Andrew. So I'm delighted now to finish up our set of talks and give a quick non-exhaustive overview on gene-environment interactions in depression. So as Luykx and others mentioned earlier, genetic factors certainly have an important influence on depression. However, environmental factors still explain the lion's share of variation. And so a natural question is how genes might interact with these environmental factors to impact depression risk. The earliest efforts involved candidate gene by environment interactions, and what I'll just quickly note here and no more is that the most recent careful evidence would suggest that there is actually no robust evidence for specific candidate genes like the serotonin transporter gene and others interacting with environmental exposures to influence depression. So what can we learn from looking not at candidate genes, but across the whole genome? This study, led by Rosanne Peterson and Ken Kendall, found that the genetic basis of depression, at least in a sample of Han Chinese women, did seem to vary somewhat by environmental exposure. They identified 3 novel GWAS hits for depression only among women who were not exposed to adversity. And SNP-based heritability was also slightly higher in this group as well, though not significantly. On the other hand, recent findings from Johnny Coleman, Jerome Breen, and team in the UK Biobank have suggested somewhat of the opposite, where heritability of depression actually appeared to be higher among individuals who were exposed to lifetime trauma, or at least reported it, compared to those with no such exposures. So somewhat discrepant findings, in large part based on very different samples, but overall suggests that the genetics of depression and what you might observe with regards to the estimates may depend on the environment that is under consideration. Other studies have used polygenic scores to aggregate these genome-wide effects and examine their interactions with stressful life events. For example, a study from NESDA found a significant gene by environment interaction where polygenic risk for depression was more strongly associated with depression in individuals who were exposed to childhood trauma. On the other hand, a study by Niamh Mullins in Radiant UK found the opposite, where polygenic effects were actually stronger in individuals who were not exposed to childhood trauma. And a later combined effort found that included these two studies as well as some others, found not much meta-analytic evidence for overall polygenic interactions with childhood trauma when it comes to depression, and that these findings may be due to chance. So this was a very careful meta-analysis, although it should be noted that the combined sample size was still pretty small. And so in the UK Biobank, which is one of the largest places that we can look right now, there have been significant interactions detected between polygenic risk and traumatic life events. So Johnny's earlier study found an additive interaction effect where polygenic effects were stronger in trauma-exposed individuals. And Shen from Andrew McIntosh's group recently reported a similar finding, again in the UK Biobank, where polygenic effects on depression were stronger in individuals who were exposed to childhood trauma, though not when looking at other later life events. So by far the most common gene-environment interactions have really focused on stress and trauma as an exposure, which makes sense, but other potential exposures have also been examined. Our group, as well as folks in the Netherlands, have looked at social support as an exposure in combination with genetic risk for depression. And what both studies have found in very different samples are main effects, where polygenic risk is associated with more depression, social support is protectively associated with depression, but these effects tended to be relatively independent and non-interactive. And similarly, when we recently looked at physical activity as an exposure, we again found independent and non-interactive effects with polygenic risk, suggesting that even if you are on the higher end of the spectrum for genetic risk for depression, as shown on the right, more physical activity or higher levels of it may still be protective against depression. And without much time to mention further, another genetically informed approach that could help us look more closely at environmental effects on depression is to also use Mendelian randomization as another way to triangulate potential causal effects of environmental and behavioral exposures on depression risk. And we recently provided an example of this by looking at the relationship between physical activity and depression using 2-sample MR. And we've also extended this to look at a range of other modifiable exposures in a paper that's currently in press. So overall, there are a lot of exciting directions for gene environment work. There are new methods that are emerging to study whole genome by environment interactions that may help us clarify or push forward or resolve some of these current discrepancies. And some groups are also taking some gene-based approaches that might help in this regard. Ruling out gene-environment correlations along the way and also addressing the heterogeneity of measurement of different environments will also be ongoing challenges that we need to keep in mind. Of course, large samples and well-powered analyses will continue to be needed in diverse samples for testing and detecting interaction effects that are clinically meaningful. So on behalf of the group here, we'd like to thank the PGC MDD, especially Cathryn and Andrew, for the invitation to present today.
Great. Thank you very much, Karmel. I'm going to give a short presentation now, just to say where we are at the moment and what our next steps are with regard to the consortia as a whole and our next data freeze. But can I ask everybody to put their questions in the Q&A box, please? And we'll come to them in a moment. I think we have a couple at the moment, and at the end of my presentation, we'll make a start on those. So you've heard from several investigators what we've been able to learn from the PGC MDD working group data and from other types of research in this field. I want to speak now about what's coming up in the next few months. So the current state of play at the moment is we have 102 significant loci identified from the paper by Howard, and there's another paper that's in review at the moment that identifies 178 loci from parallel work. And from the Howard paper, we've identified in 807,000 individuals, or almost 250,000 cases of people with depression. And to do this, we've adopted a spectrum of phenotyping approaches, from minimal single question phenotyping to electronic health record to interview-based phenotyping, or more conventional approaches. And we've used this to expand our sample sizes whilst trying to thoughtfully achieve our goal to maintain and increase clinical relevance of our findings to people who are most in need of genetic and other discoveries. So what's coming next? Well, we expect our next data freeze to be 1 month from today. So we have uploaded or in process European ancestry samples from 4.3 million individuals and more than 400,000 cases. This includes 23andMe data, which has been sent to us today. And we expect to set the next data freeze 1 month from today, from the receipt of that 23andMe data. We've also a trans ancestry data set of more than 750,000 individuals and 32,000 cases of Latino, African, and East Asian descent. So what I want to say to you now is that if you're listening to this call and you're not already involved in the MDD group of the PGC, then please do reach out to one of us and become involved by going to this web link here, and you'll find details of Cathryn and my details and the details of other people who can help you. That can be in respect of contributing new samples and being part of the next major data freeze and GWAS meta-analysis, or it might be in conducting your own secondary analysis proposal of existing data. If you're a member of the PGC, you can apply to do one of those secondary analysis proposals directly. And if you're not, you can reach out to a member of the PGC in order to do so, and we will help you do that. What we want you to appreciate is that we are very welcoming here, and we would very much like to work with you. And this is a picture of where you could be in a few months' time. And, there are extra points here for people who can guess the identity of the person whose face has been obscured. So thank you to all the speakers today. It's been a really interesting set of discussions, and it's now open for questions.
So we've got one question in the Q&A from an anonymous attendee. I don't know if you want to give the question yourself, or shall I just read it out? It's asking whether it would be helpful to tackle the wide range of the MDD spectrum, which we've heard a lot about from several of those talks, by starting to assess polygenic risk score for various traits related with MDD rather than diagnosis, and see if a combination would predict better than the MDD subgroups. I don't know who wants to take that. I mean, it perhaps comes into your talk, Jurjen, on the genetic correlations, but anyone else that has any comments about how we're going to cut through some of these challenges of the MDD spectrum would be very welcome. Anyone? No?
I think it's one of the probably most clinically useful applications of genetics in MDD is to try to use polygenic risk scores combined with other measures to identify more homogeneous subgroups in MDD that can then be, for example, treated better. So I think, yeah, this is a great idea, and this is, in my opinion, what will most likely be useful to as an application of genetics to optimize treatment of MDD.
Great. Till, anyone else to contribute? I mean, my perspective on MDD, I think, is that it's always going to be difficult, as we've heard both the prevalence and the low heritability both in a way work against us with the lack of a very clear-cut definition. And I think we're going to have to try all the different approaches that we can. There's not going to be a single approach that's going to be, the best way to detect these loci, to figure out what's going on in the biology, and to move that forward to helping patients. And so these types of approaches of using polygenic risk scores could be really valuable. Please do add your Q&A, your questions to the Q&A box. I know Pat has two. Do you want to ask those yourself, Patrick?
Yes. The first question is, so the yield of candidate gene studies has been demonstrably very low, if not, in fact, actually zero. Why do we still see candidate gene studies? I get asked to review a candidate gene studies, a couple times a month. And why do researchers still refer to the disproven literature as being something worth looking at? So for example, if you talk to some people who aren't like on this call, they'll tell you that the serotonin transporter, for example, is a proven G by E factor for major depression.
So from my perspective, there are probably a number of reasons. So one is that I think some people still have this sort of very romantic notion of we can identify one genetic variant that then can actually, that has like a known implication in the neurobiology of the disease that then basically can trigger this whole cascade down, which will understand the underlying biology and will actually be able to develop drugs and so on and so forth. Although that is probably not something that is very realistic and probably shouldn't be performed anymore, considering that it just seems like sort of a waste of money and resources. But I was also very interested to see or struck to see that the study that you're referring to is actually one of the most highly cited papers there are in psychiatry right now, with over, I think it was over 8,000 citations. And I did wonder about that a little bit as well. But maybe Karmel, you want to add something to that?
This is referring to the Caspi et al. Paper in Science 2003, the serotonin, the CERT polymorphism, the HTT-LPPR thing, which they claim to have a gene-environment interaction. There's been a super large meta-analysis of that, which has come out completely negative. But yet one still sees this, and it is, I think it's the second or third most highly cited paper that the NIMH ever funded, and it's wrong.
I think just to add to what Eva says, I think that because the genetic variant occurs at a gene encoding the site of action of SSRIs, I think people just find the biology just so seductive, don't they? I think people want there to be a simple answer, and I think often science is about finding the simplest explanation for things. It's just that in this particular case, it doesn't work, and people find it very difficult to unlearn those habits, I think.
I agree with the biological plausibility angle and how that really draws people to continue to try to explore those relatively simple and seemingly elegant questions that you can look at. I think with regards to the National Institute of Mental Health in the US, there is that movement to really review, to not consider a lot of these candidate gene by environment interactions anymore in terms of funding priorities. So I think there is a move in that direction, at least at that level.
And just to finish off on that, unless anyone else has anything to say, I think there's also a practicality here that for people in small groups where resources are a real issue, doing GWAS is challenging, but doing genotyping a few candidate gene SNPs is a potential research study that people can do. And I think that PGC perhaps has a role to play here. Andrew talked about the possibilities of getting involved with large data analysis, getting involved with the data sets that we have available, a lot of public data sets outside of what's in the PGC. And I think perhaps we have a responsibility to suggest, well, let's think about what else people can do that will really contribute to global science, alongside what they're able to do in their small research group. Anyone else want to add to that question?
Yeah, can I?
Yeah.
Just like Karmel mentioned, I think it's just quite a lot of gene environment interaction studies there. This topic has been suspected. However, the Caspi paper in 2003 is just the most highly cited or most high-profile paper out there, and people just referring to this paper with this suspected gene environment interaction. Anyway, I just wanted to say that it highlights how important it is to have to run a really well-powered and clever design study on gene environment interaction.
Okay. So we've got one question from John Nuremberger in the chat, and then I'll come back to you, Pat, for your second question if we have time. John's asking, and perhaps this is one for Till and then Andrew, as we're now approaching 178 loci in GWAS, what's the end game in these studies?
I think at some point it will not be about counting the number of associated loci anymore, because that's anyway arbitrarily based on a p-value threshold, how many you have. But I would assume until a certain time and sample size, polygenic risk scores will get better, because the effect sizes are actually estimated more reliably when the sample sizes increase. So I think at some point we will have to go away from counting the associated loci, but see that all the downstream analysis, from pathway analysis to polygenic risk scores, just get more reliable with larger sample sizes.
So I just add to that, Cathryn, that I don't think this problem is going to trouble us particularly soon, but I think it will be at the stage where we explain the majority of the SNP heritability for depression. I think I don't think we necessarily need to explain it all, but I think if we explain a big chunk of it, we can then not only identify and help stratify people's risk using the polygenic risk score in a way which is clinically meaningful, we can also maybe start to adjust for genetic factors in studies of other important environmental variables also. And I say, to do that, I don't know what percentage we need to, but I would think that it's somewhere of the order of 3/4 perhaps of the overall SNP heritability. I would think we might start to get close to where we need to be.
But if you want to have some controversial discussion now, and do you believe that if we increase the sample sizes, identify, so to say, more genome-wide significant SNPs and so on, that we will really get there, that for MDD we can get PRS that on a clinically relevant level, that reach sensitivities and specificities that they can be used for risk stratification in a meaningful manner? Do you think that will be possible?
I do. So I do think that, and I don't think we're that far away from having something which is clinically meaningful, because you don't have to. So if you go back to the original papers where people were sorted into deciles, and you looked at the very highest decile of people, now a polygenic risk score might, in some circumstances, might not be relevant for the majority of people in most deciles. But if the highest decile has a relative risk at the moment of 2 to 3, and we might get to 5 to 10 or even greater in future, then that might change the interaction clinicians have with patients who are in that highest bin of risk, and bring on top of that information from exome sequencing or information from one of our subgroups, the antidepressant treatment response subgroup. One can see that that information feeds into the clinical consultation and can be brought to bear on how patients are managed in everyday clinical practice.
Anyone else?
Can I throw in something real fast in that too? So I'm going to put a paper in the chat box in a second, but we've had 2 papers in Nature Genetics now where we've argued that the readout of GWAS is actually identification of brain cell type. And I would ask people to look at that, because that might be actually what's going on. That might be what the GWAS are telling us.
Anyone else of the panelists want to comment on that? Where next for GWAS? No. Then Patrick, can I ask you to ask your last question, your second question in your box, which I think is probably quite a good one to finish with.
Yeah, so it occurred to me that we're in a situation for a horrible natural experiment, which is the opportunities that the pandemic, the COVID-19 pandemic, may offer for gene by environment studies in major depression. Obviously, this is a widespread but not universal exposure with multiple different fear, anxiety, and real and imagined impacts on people. Does anyone care to comment about the opportunities there?
Since I talked about gene environment stuff, I'm happy to jump in and curious what other people think as well. So definitely COVID-19 serves as a widespread stressor and has clear impacts on mood and depression, among many other mental health impacts. And like you said, it does serve as somewhat of a natural experiment that affects a lot of participants who have been in these cohort studies that we include in our GWAS and our genetic studies. So we already have genetic data on many participants who, are living through this experience. And so it does offer some interesting, unfortunate, but interesting opportunities to look at gene and environment interactions. I think the environment piece is something that we need to be thoughtful about in terms of how best to operationalize, because it is so widespread, but there is also variability in terms of actual exposures and stressors within the kind of pandemic experience, whether loss or work-related stressors or financial stressors, and also the degree of perceived stress. So I think there's just a lot of different ways to characterize the E part of the equation that if we were to sort of do this in a systematic and thoughtful way, I think could combine really interestingly with the genetic data to answer some important questions.
And I think it's great to see that some of the psychiatric case control cohorts, they were very quick to develop extra interviews done by telephone, for example, to assess some really specifically how the COVID-related stress affected their participants in their depression case control studies. So I think these information will be really useful then to gain some insights in how this affected people.
Yeah, I could just to add to that. This is an opportunity, I think, for us to learn things about depression we didn't know before. And again, the field has come together and actually decided to use many of the same instruments across many different countries. And so many people on this call will realize that the ability to compare results across different countries of similar ancestry has been made very difficult by the fact that we often include different scales, different measures and definitions of depression across those countries. And so now we're faced with actually quite an opportunity to make comparisons across different national governments and systems, and to maybe learn a bit more about depression than we'd ordinarily be able to.
Can I ask, is there any plan for PGC to form a group to look at the G by E in this case? I mean, we might have the opportunity to have a better G by E profile paper than the balance rate.
Are you offering?
That's a really good idea. If you'd like to take that on, Louis.
Well, I would wonder, what about raising this, you know, maybe doing 10 or 15 minutes on it at the next MDD call to see whether there's, you know, a collaboration of people that might want to put their data together at some point. You know, typically in the PGC, you write your own paper first. Like, I think, Andrew, you guys plopped something onto bioRxiv or medRxiv last week, right? Or 2 weeks ago. Can you put that in the chat box too, so people can see that? Okay. But at some point, you know, you'll learn what you can, and then the next step is to go to a meta-analysis to basically pull it all together. And it might make sense to see whether there are people that have comparable data. If there are enough, then maybe there's something to do there. But if there isn't, then there's nothing to do.
Great, so we have no more questions. I just want to highlight a comment that Palmer's put in the chat, that reminding us that part of COVID stress is the social isolation, which is an environmental source of loneliness, but which of course is not identical to depression. And as we've said, I think there's going to be a lot of dissecting out, as Karmel said, what's going on here.
So I just one comment on to response to Abe real fast. So part of that depends upon personality type too. Because some of the people that have been kind of the most pleased with this pandemic and being quarantined are people who are quite introverted who have a partner, right? They actually, many of them have told me that this is actually, one of my, I know someone whose partner said, quote, "My whole life has been in preparation for this moment." Because she's, for example, very pleased with the fact that she can do everything from home.
And maybe scientists to finally find time to write their manuscripts.
I think you mean
they're at home with children.
Yeah, I think you mean male scientists, right?
Yeah. Yes. Right. I can see we're on the hour, Andrew.
No, I was just going to say that might be a good point to close. I mean, I think we all agree with you, Pat. I think there's much to be done in this space. But I think probably now's the time to thank everybody who's joined the call for dialing in and listening to these early career researchers. And thanks most of all to the early career researchers for coming and giving their talk. So yeah, bravo. I think probably now is the time to close the call. So thanks again, everybody, and cheerio.
Bye, thank you.
Thank you. Bye, thank you.